College UK Cambridge Centre podcast. In this Integrated Cancer Medicine Research in Focus series, I talk to various ICM members about their research and how it is supported by the vision of the Mark Foundation Institute for Integrated Cancer Medicine. MFICM research uses cutting-edge analytics to maximise the use of diverse high-volume datasets and by capturing cancer heterogeneity in time and space in patients receiving active treatment. Integrated cancer medicine aims to transform the way the world treats cancer by affecting patients along their treatment pathway and ultimately accelerate cures. Today I have with me Professor Kevin Brindle, and we're going to talk about his life's work in imaging research. Kevin is Emeritus Professor of Biomedical Magnetic Resonance in the Department of Biochemistry and a Senior Group Leader in the CRUK Cambridge Institute at the University of Cambridge. He is also a Fellow of the Academy of Medical Sciences and of the Royal Society. So good afternoon, Kevin. Thanks for joining me.
Hi.
To start with, please could you tell me what your research focuses on currently?
Pretty much since I've been in Cambridge, which is over 30 years now, I've really focused on using imaging to detect very early treatment response in cancer. And currently that involves metabolic imaging using hyperpolarized C13 labeled and deuterium labeled substrates. The C13 we've taken to the clinic already, and deuterium we're in the process of doing that at the moment with new probe molecules. We're also taking to the clinic an agent I've worked on for over 20 years. It's a PET agent for detecting cell death. We've now made that under GMP. We've worked out how to label it with F18 for PET, and we also plan to take that into the clinic in the coming year. And then the other area that we're currently working in, and we've worked in for some time, is a novel gene reporter allowing us to track cells in vivo. And in the collaboration with Michael de la Roche in the Institute, we're using that to track CAR T cells. Currently in model systems, but ultimately we think that could also translate into the clinic.
So you've used loads of terms and acronyms there that my listeners may not necessarily understand. So
Okay.
what we'll do is we'll delve a little bit deeper if that's okay with you. And if you can just explain to me what these things are doing, what they're showing you and how they might impact treatment and why it's important to get them from the research bench into the clinic.
Okay, well, if I start off with metabolic imaging then, we've taken into the clinic some time ago now, hyperpolarized C13 labeled molecule. Now, magnetic resonance is a very insensitive technique, and this method of hyperpolarization increases the sensitivity by about 10,000-fold. So we've worked on this for the last 20 years. We were amongst the first to use this technique. And essentially what it allows us to do, we can take a metabolite, a cell substrate, something that cells take up and use to make them grow and produce energy. We label it with this carbon-13 label, hyperpolarize it, and then we inject it intravenously. And we have so much signal now that we can detect where that molecule's gone in the body. And most importantly, and this is a feature of magnetic resonance, is we can look at the conversion of that molecule into other metabolites inside the cell. And the rate of that conversion tells us something about the behavior of the tumour. And a particular focus, as I said, is we wanted to image treatment response to get very early evidence of whether a patient's tumour is responding to treatment. And these metabolic changes can be very, very quick. And we're currently in a collaboration with AstraZeneca. And for the first time in the clinic, we will use this technique to image response to an AZ drug in breast cancer patients. And we think we'd be able to tell whether they're responding within a few days of starting treatment, whereas conventionally it can take weeks or months before the patient knows or the oncologist knows whether the drug is actually working. So I think, we talk a lot about personalized medicine. One way to do that is to upfront, say, sequence the tumor DNA to get some indication of which drugs might work. Complementary to that is this approach where when you start treatment, you find out very quickly whether indeed that drug is actually working.
And that's really game-changing, isn't it? Because if it isn't working, then it will stop that patient having all the extra toxicity in their body that continuing, that treatment would give them.
Yeah, and people often say to me, oh, well, imaging is very expensive. Well, I think you need to put that in context. These drugs are extremely expensive as well. And if you're treating somebody for a year with a drug that's not working, that maybe cost you $100,000. The cost of the imaging is trivial in comparison with that, for example. So there's benefits for both the patient and for the healthcare system as well.
Yeah, I can absolutely see that. And when you say a hyperpolarized molecule, are we talking a radioactive molecule?
No, it's not right. No, so magnetic resonance doesn't use ionizing radiation. Carbon-13 is a stable isotope of carbon. Typically it's carbon-12, but carbon-13 is about 1% naturally abundant. And you can detect it by magnetic resonance, but it's very insensitive to detection. So this hyperpolarization technique radically increases sensitivity. And if you think of what you're doing in a magnetic resonance experiment is that these nuclei behave like, if you like little magnetic dipoles. And they line up in a strong magnetic field. And we can detect that alignment. This hyperpolarization technique makes them almost all line up with the field rather than a few parts per million lining up. And that gives you a massive gain in sensitivity. Really quite unprecedented. What we were actually working on more recently is using deuterium, which is an isotope of proton, hydrogen. In this instance, instead of using carbon-13 as the label, we use deuterium. The advantage of using that is hyperpolarization requires quite a complex and expensive piece of kit to impart this hyperpolarization. With deuterium-labeled substrates, you can literally take them off the shelf and inject them. And in fact, what we're really excited about is, as well as intravenous administration, what's actually more attractive is that they can also be administered orally. So the patient would just drink this labeled material. And again, no ionizing radiation. And a lot of the molecules we've used have been used already in patients, non-labeled, but have been used in patients. So translation into the clinic is really much more straightforward. And it's again looking at pretty much the same thing. We're looking at tumour metabolism. As I said, tumour metabolism is frequently aberrant. Tumours need to grow rapidly, or they do grow rapidly. And in order to fuel that proliferation, you need to make lots of biosynthetic intermediates. So we can image that process effectively. So the faster they grow, they use more nutrient, if you like. And so that gives us a way of detecting tumours. And as I said already, when we hit them with a drug that's killing them, then that all slows down and you can see that.
Another term you used was GMP manufacturing. Could you just expand on that a little bit?
Yeah, so that's manufacturing in a form that allows us to then inject it into a patient. So I was referring there actually to another agent that we've developed. This is actually a PET agent, positron emission tomography. This agent is labelled with a very short life isotope of fluorine 18. It's radioactive, it emits positrons, and positrons are anti-electrons, they're actually antimatter. I don't think people going into the clinic realise they're being imaged with antimatter. Positrons were really predicted by Paul Dirac back in the 1930s. It's now a commonly used technique in the clinic, FDG PET, which is a glucose analog, a fluorine 18 labeled glucose analog. What we've done is put this fluorine 18 label onto a molecule that binds to dead cells or dying cells. And so we can image cell death. It's been a long-standing desire for people in imaging to be able to image cell death. And actually, remarkably, given a lot of effort, we don't really have a good way of imaging dead cells in tissues in the clinic. And we think our agent stands a good chance. We've worked on it for many years and now got it to the point where we can take it into the clinic. And so again, it's the same idea of imaging early treatment response. So if you treat and you get lots of dead cells early post-treatment, that's a good prognostic indicator. That's an indicator that the drug's working. And depending on how much cell death you see, that may give prognostic indication of longer-term outcomes. Or at least that's the idea anyway. So it's been a bit of a passion over more than 20 years now to get it to this point.
Absolutely amazing. And what an amazing idea and cutting edge research. And it's so exciting to have that now pushing forward into the clinic. I mean, how wonderful. Let's go back to the beginning for you. Where did you start your career and what inspired you on this path in the first place?
I was at the University of Oxford as an undergraduate doing biochemistry. And I was really interested in structural biology, actually. This is back in the 1970s, determining structures of proteins. And it was a 4-year course in Oxford. And the last year is a sort of what's called a Part 2 course, similar to actually Cambridge, where you get to do a research project. And I was really interested in, as I said, in structural biology. I was really interested in NMR because we'd had lectures on NMR, and I really didn't, it was the one bit of my course I thought, I don't really understand this. So I'll go and do a part 2 project to see if I can better understand how this technique works. And I was interested in structural biology anyway. So I went and did a part 2 with a guy who turned out to be my PhD supervisor eventually, a guy called Ian Campbell. And when I arrived in the lab, he said, oh, no, I think structural biology, well, he transiently thought this. He said, oh, I think, we can't determine structures of proteins by NMR. This is in 1977. I'm going to drop it. We'll do NMR on cells. And that's how I got into this whole game. So my part 2 project turned into a publication. And in fact, that paper is still cited even to this day. It turned out to be quite an important paper. I mean, I thought, oh, this is really quite interesting. And so I sort of stayed on with him, did a very short postdoc, and then worked with another guy called George Rada in Oxford. And then I got a Royal Society Research Fellowship. So it was all accidental, really. It started off me being interested in the technique and wanting to learn more about it. And I was interested in structural biology. And then I got completely diverted into actually looking at tissue metabolism. And I have to say, as an undergraduate, I was completely uninterested in metabolism. We thought, this is all old, So Hans Krebs was still working in Oxford at that time when I was an undergraduate. But we thought, this is old biochemistry. We don't need to do this. And so I really wasn't that interested. So it was all accidental, really, how I got into it.
It's amazing how that happens, isn't it? And then suddenly you're off on a tangent and suddenly it's all really important.
Well, that's right. It's and that's 40 years ago now.
Wow. How long have you worked at Cambridge University for?
31 years. I came in 93.
Wow. And it's a really hard question, but can you tell me what your highlights have been since you've been here?
Yeah, so I think what really kicked off the whole thing with CRUK was I was working with a guy I'd met when I was in Oxford, a guy called Jed Adams, who was head of MRC radiobiology unit at Harwell. Again, it's sort of accidental. They lost their NMR spectroscopist out at Harwell, and they asked me if I would go and help out. So every Friday I would drive down and help them do this imaging work. What they were working on was an antivascular drug called combretastatin. And I said, well, I could do this actually in Cambridge. I could image whether this drug is actually working. It shuts down tumour blood vessels. And so we devised a way of imaging using contrast media, which are used clinically, to detect very early again whether this drug was working, whether it was shutting down blood vessels in these model tumours. And that was so successful that went to the clinic. So that was my first taste of this. The techniques that we developed to detect response in the lab were then used in a clinical trial of this drug. And I think that was probably the first time that had happened. So I saw very early on the potential of doing this work and also of translating it into the clinic.
And you just sort of carried on in the same vein?
Well, yeah, pretty much from there. As I said, I was really interested, back in Oxford, I'd worked on metabolism, actually using proton and phosphorus spectroscopy. And I'd sort of dropped it because I thought this is going nowhere. The sensitivity is too low really to do anything sensible with it. And then it was 2003, I was speaking at a meeting at the Institute of Radiology in London, and a guy called Claes Goldman stood up and presented this new technique, this hyperpolarization technique. And I just sat there thinking, this is amazing. So I went straight down to talk to him at the end of his talk and said, I really want to do this. This is fabulous. And so we got talking, and then I went out to Malmo in Sweden, where they were based. And kind of long story short, we ended up getting a prototype polarizer in Cambridge in biochemistry in 2006. And so my interest, whereas my interest in metabolism completely drained away because I thought, this is just not sensitive enough, was completely changed then by this new technique. So we jumped into it, and I pretty much devoted the whole lab at that point to this. And we published some what became quite important papers. And so that was certainly an important step in my career. The other important step was development of this PET agent, which is now going into clinic.
You must have had lots of collaborations with various different people. So who have your most memorable collaborations been with, both inside and outside Cambridge?
After I left Oxford, I spent 3 years in Manchester. I actually had a really nice lab set up in Manchester, but I left because I thought the potential for collaboration would be greater in Cambridge, and that indeed has turned out to be true. So in total, I've collaborated with 5 people in biochemistry. These are published collaborations, 8 people in the CI, and a total of 32 external collaborators, all with people we've published with, and 16 of those are in Cambridge. There's a lot of names here. I mean, it's hard really to pick out any one individual because they've all been in very different areas. But it's been fabulous being in Cambridge because you're surrounded by lots of smart people. And if you want to do something, you can find somebody who can help you do it. And that's certainly been my experience.
Yeah, I think the breadth of possibility for collaboration in Cambridge is huge, isn't it? Just within the university, then, tell us where they've been.
We're going back over 30 years now, so it's been quite varied. So, for example, in biochemistry, I worked with a guy called Martin Brand on, because I worked on yeast actually a while ago, and we worked on mitochondrial and coupling proteins. Martin's actually a very well-known mitochondrial person, and we worked on that together. In the CI, Carlos Caldas, we were working on breast cancer models, Dave Tuverson on pancreatic cancer, Florian Markovic. I had a collaboration with a guy called Mike Hobson, who's an astrophysicist. We were working on image analysis. This is pre-radiologics time, so I worked with Florian and Mike Hobson. Mike Hobson, as I said, is an astrophysicist. David Neal on prostate, and Nitsan. We published quite a lot with Nitsan. I mean, people started teasing me when ctDNA came out. They said, we don't need imaging anymore. We can tell whether the patient's responding. Just look for tumour DNA in the circulation. So I got together with Nitsan and said, let's see if this is really true. And so we published, mainly work driven by Nitsan, I have to say, published quite a lot with Nitsan. More recently, Tim Halen, we did some mass spec imaging with him. I think of people in Cambridge that have been really important, and I've published a lot with, Finian Leeper, who's an organic chemist. I mean, we published a lot of work on imaging cell surface glycans using what's called click chemistry, and all that chemistry came from Finian. Finian was a brilliant collaborator for me, actually. We had a really very productive collaboration, published many papers with joint students. Robin Franklin, I've collaborated with for years on cell tracking, using this gene reporter that we've developed. We're about to publish our final paper, I hope in the next month or two. Bazbek Davletov in the LMB. So this agent for detecting cell death, that came out of a collaboration with Bazbek. So Bazbek is an X-ray crystallographer, at that time working in the LMB, and he worked on this protein called synaptotagmin. And synaptotagmin binds to this target that's exposed on apoptotic cells, on dead cells. So that's how this agent came about. He had all this expression system all set up. So we were able to import that directly into the lab, make this protein, and then show that we could use it to image cell death. So that was a very important and very productive collaboration. Rebecca Fitzgerald, we worked a lot with Rebecca on, and this is work that subsequently Sarah Bondic, who did a postdoc with me, has worked on. That's it, using fluorescently labeled imaging agents to detect tumour cells in the esophagus. We published with Rebecca on that. Colin Watts, we started the work on brain tumours actually with Colin, and I'm working quite a lot now in brain tumours. And some of these clinical trials that I've been talking about will be in glioblastoma patients. And that will be with Richard Mayer. So Richard did his PhD with me, and he's now in clinical neurosciences. Actually has quite a big lab there now and is continuing with that work. So we'll collaborate with Richard on that. So yeah, pretty diverse range of people actually.
It's just amazing to hear the breadth of the collaborations and the amount of people that you have influenced and have influenced you over your career. So thank you for expanding on that. And I know, having spoken to Stephen Price recently on a podcast, how important imaging is in brain tumours, because of course you can't get to them to take biopsies. So that ties in, yeah. Who has inspired you? I think you've inspired many people. Who inspired you?
Yeah, I thought a lot about this. It actually goes back a long, long way to when I was at school. So at school, I did, for A-levels, I did biology, chemistry and maths. And to be frank, I thought the biology was so boring. It was naming of parts, naming bits of flowers. I thought, I don't really want to do this. And obviously, we were taught about classification, Linnaean classification. Now, I recognise the importance of that, but I thought this is so arbitrary and it doesn't tell me how anything works either. Whereas chemistry, I thought, there are rules here. I can see how things work at a molecular level, which is why I was really interested, which is why I was interested in structural biology. I vividly remember it because for some reason, we were in a different classroom to what we were normally. And the teacher started he talked about how the triplet code was solved, the triplet code for amino acids. And I thought, this is chemistry. And this is chemistry that explains how biology actually works. I thought, this is brilliant, because up to that point, I thought, I hate biology. This is so boring. Well, certainly A-level biology, as it was taught back then. This was in the late 1960s, early 70s. And then all of a sudden, I thought, this is not only here was a molecular description of how some biological system actually works, fundamental chemical understanding, but this is still going on down the road in Cambridge. So I thought, this is what I want to do. I want to do biochemistry. This is what I'm really interested in. So the people who inspired me were people like Sidney Brenner, Francis Crick, these heroes really in the field. I would never have done biochemistry, but for that lesson actually, because I thought, I don't want to do biology.
So that wonderful teacher who just set your mind alight.
Yes. The biology teacher I didn't think much of, apart from that lesson. The chemistry teacher I thought was fantastic.
It's amazing how one teacher can influence you in that way, though.
Oh, yeah, no, my chemistry teacher was amazing, yeah.
How do you think the landscape has changed in imaging research since you started your career? This may be a long answer.
No, I think that what really has changed quite fundamentally is just the hardware is so much better now. And mainly, actually, that's because a really important aspect of that, which we almost take for granted now, is computing power. When I started, the computers you had to bootstrap them by literally dialing in numbers to set it going. They were unbelievably slow, very cumbersome. The whole machine was built around the computer, actually. The computer was a major part of the expense of buying an NMR machine. Now, of course, you could run the whole thing off a laptop. And to put it in context, I was at a conference a few years back, and somebody was talking about image processing. And when I was an undergraduate, the big computer was the Cray supercomputer. It occupied multiple rooms, dimmed the lights when you turned it on, this really heavyweight computing. And this guy said the iPhone 4 had more computing power than a Cray supercomputer, to put it in some sort of context. And so, we take it for granted now, but I can remember walking around with disks this big for 256K of data. And now I've got gigabytes in my pocket, or megabytes. So that's made an enormous difference. And it's allowed us to do experiments that simply weren't possible. I can remember as a postdoc, I wanted to do a complicated pulse sequence in magnetic resonance. And I had to buy a new pulse programmer which allowed me to do just 16 steps. Now you have programs with hundreds of steps in them. So I think that's probably, everything's got better. The hardware has got better, but I would say probably computing has been the biggest change that I've seen.
Yeah, it's staggering, the rate of change in that department. It just is exponential, isn't it, seemingly?
Yeah. And we sort of take it for granted now. And that's probably, I would say, the thing that's changed the most. Basic ideas of what we're looking at and the techniques we used haven't changed, I guess, that much. I mean, one of the things about magnetic resonance, my PhD supervisor was a physicist. He was doing structural biology. It was really him who instilled in me this love of the technique. Because what he said, the theory for magnetic resonance is just beautiful. I mean, it goes back decades. And, I would say I'm no expert. My knowledge of magnetic resonance is quite superficial in places. It's quite a complex technique, and it's very broad in terms. But I understand what you can do with it in biological systems, having worked with it for so long. But really is an elegant method. Its biggest problem is it's so insensitive. That's what we've battled with all through, is lack of sensitivity. That's why this hyperpolarization technique was such a revolution in terms of what we could do.
How do you think your work has impacted cancer treatments? You've talked a bit about things going into the clinic. Are you there yet, or is it to come? Or
no, so somebody I know very well, quite a well-known figure in the field, I won't say his name, but he once said, metabolic imaging is a very promising technique, and it always will be.
Nice.
So what I would say is, yes, we've had 40 years. We know pretty well what it can do, and it has been used in the clinic. And in some places, actually, spectroscopy is used routinely, but it's not used routinely and widely. And there are a variety of reasons for that. I still believe that the technique has that potential. I think for therapy monitoring, and we'll find that out in the next few years, just how useful that's going to be. I think part of the problem with I'm talking about magnetic resonance, the PET agent, if it works, will go into clinic. There's no question. I mean, the infrastructure's all there. But metabolic imaging, which has really been, I suppose, my passion for 40-odd years, it's quite challenging to do. But I see incrementally we're getting closer to making that a reality. People don't like this term, but I think it's pretty apt. Killer application. Something that clinicians can say, yeah, I need to know this in order to treat my patient. And we're not quite there yet, but I still believe we can get there. We've made a huge amount of progress. I mean, when I started with the carbon-13, I shared a platform very early on with somebody who said, I'll never go to the clinic. But it has. Now, that's one thing. Whether it'll do anything useful in the clinic, I think it will. We've demonstrated the potential, the capability and what you need to do. And I think in the next few years, we'll find out whether it can do that. But yeah, I think if you could tell whether a drug's working within days of starting treatment, that has to be a useful thing to be able to do.
Yeah, that has to be game-changing, doesn't it, really? Where do you see integrated cancer medicine taking us in the next 5 to 10 years?
Yeah, I see what we do as sort of complementary. Really looking at ctDNA, for example, you might see a spectrum of mutations that might then indicate which drugs you're going to choose. And so I see what we do as complementary. So we say, yeah, okay, it is actually working. If you've got multiple tumour deposits, is it working the same in all of those tumour deposits? You can't biopsy them all. For example, in ovarian cancer, you can't biopsy them all. So imaging is one potential way of giving you that overall picture of everything that's going on. So I think I see them as complementary. I think I would like to dispel this notion that imaging is always expensive. As tests go, it's not that expensive. I agree for the C13, it is pretty expensive right now. I think if it was expanded and used a lot more, then potentially those costs could come down. But as I said earlier, you have to put it in context. These drugs are expensive as well.
Where do you see imaging research taking us in the next 5 to 10 years?
What we've demonstrated so far, based on preclinical work, is what these metabolic imaging techniques can do. And I'm speaking now for metabolic imaging. I'll talk about maybe other MRI techniques in just a moment. What we really need to do is now well-controlled, multi-center clinical trials to demonstrate real utility, to get some evidence base that this is actually useful clinically. Now, those are quite tough studies to do. They're not particularly attractive to do if you're a scientist, actually. Just to put that in context, so our PET agent, first paper was published in 2001 in Nature Medicine. The last paper that we published on the fluorine-18, which is what's going to go into the clinic, is published in a journal that nobody in the CI will ever have heard of. So it becomes pretty unglamorous at that stage. That grunt work of actually getting into the clinic is actually, you're not going to get, the high-profile publication was the first one which showed the potential, then actually getting it there is a lot more challenging. I think that's really important, that focus on clinic now to demonstrate utility. In general, for other techniques, again, actually, if we just take spectroscopy, metabolic, not polarized C13 or deuterium, I think if you look at in prostate, spectroscopy's worked there, proton spectroscopy has worked, but it's not widely and routinely used because it doesn't add enough to what's already available. But I think if you look at, people talk about a lot about early detection. You don't often think about MRI as being a great technique for early detection. But actually, if you look at the prostate, what you want to do is detect a lesion that's going to cause you a problem at a point when you can usefully intervene. And I think in prostate, you've got patients with lesions in their prostate. I think MR now is getting to a point where you can tell whether those lesions are a problem or not. So I think that's an example of actually early detection. The lesions are already there. It's just telling you whether this is a lesion that you need to worry about. And if that gets rid of biopsy, that's really quite powerful. I think these things are all complementary. The MRI is relatively quick, 20, 30 minutes, and you had a blood test as well. And that would tell you whether this is a lesion you need to worry about in prostate. I think that's important because we know that prostate, men are being overtreated. Men are getting treated when they don't need it. So I think there are examples like that which are really quite important. And of course, if you want to know what's going on, MRI has been used for a long time.
Once you leave your university post, what are you most looking forward to doing?
I'm not stopping work completely. I mean, that would be like running off a cliff, and find nothing underneath you. I have a consultancy and membership with 3 SABs in Germany. So I'm spending quite a bit of time in Germany. And there's something else that I'm sort of developing at the moment in Germany. So I spend a lot more time there. Part of the attraction is I have a new grandson who happens to live in Germany. So that's an opportunity to go and see him.
That's perfect.
Yes.
Well planned.
But outside of work, but I do a lot of cycling and gardening. I'm still working it out because it's quite a big change. I mean, this whole thing of coming up to retirement, I definitely wanted to do something different. I mean, I've run a big lab, been there, done that. I didn't want to just carry on doing that. Equally, I didn't just want to stop completely. So finding a sort of middle way through, it's quite a difficult change because in a career in science, you just go after everything. And now in the last year or two, I think, well, I can't do that now because I haven't got time to do that now. I can't take a PhD student to work on that because I haven't got time. So that's quite a big change in mindset, really.
Things that you've been working on for 30 years are suddenly now, as you're sort of finishing your university career, are coming to fruition and coming into play.
Yeah, well, that's quite sort of gratifying in a way. But people sort of say, well, surely you want to carry on doing that. There'll always be something new. There's always something new. That'll always be the case. You would never stop. You could never stop because there'll always be something new to do. So, I'm not really that bothered about that, actually. I mean, I'm really pleased that these things are going into the clinic now. They either work or they don't. I mean, let's be frank. It's an experiment. We're trying it out for the first time. We're pretty confident that, we know that they'll work. Whether they give us anything useful in terms of patient management remains to be seen. But yeah, I'm pretty happy with where it's got to at this particular point. I guess if it had been a few years ago and they hadn't gone into the clinic, I'd be quite frustrated by it. But I'm not frustrated by it. It seems quite a good place to wind down, not stop.
If you were to give one piece of advice to the next generation of metabolic imaging researchers, what would it be? Focus
on translation into the clinic, because we work on length scales for MR on the millimetre sort of length scales. Hard to answer sort of fundamental biological questions at that sort of image resolution. Biologists typically want to work on the micron scale. But when you take it into the clinic, you can start to do things that are really not possible using other techniques. In terms of models, all models are bad, but some are useful. Well, if you go into the clinic, you don't have to work on a model anymore. You're working on the patient with this technique. So I think that capability to do studies in patients, and it's really sort of almost a unique capability as well with magnetic resonance. For looking at metabolism, you can do PET as well. So I think do fundamental research, but translate that fundamental research into clinic.
It's been so interesting hearing about your amazing career this afternoon, and thank you for taking the time to talk to me about it. I wish you all the best for your retirement.
A pleasure. Thanks for inviting me.
If you want to find out more about the work of the Mark Foundation Institute for Integrated Cancer Medicine, please visit our website at www.integratedcancermedicine.org, where you can find details of the ICM vision, all the current research, clinical trials, resources, publications, and team information. You can keep up to date with our latest news and events, and you can also sign up for our newsletter. If you would like more information about the work of the CRUK Cambridge Centre, please go to www.crukcambridgecentre.org.uk, or you can connect with us on Twitter using our handle @CRUKCamCentre. Thanks for listening, and do join us again soon.